I am thrilled to introduce Nathan Pethick. Nathan recently graduated from Oregon State University with an honors Bachelor of Science in biology, specializing in ecology and also minoring in chemistry. As part of his undergraduate studies, Nathan completed an honors thesis titled Testing the Response of Mid-Intertidal Prey Communities to the Mass Mortality of a Keystone Predator, Pisaster ochraceus or ochraceus. It was up for debate. I asked Nathan how to pronounce it, and he said it was either way. This research, done right here in the Cape Perpetua region and mentored by Dr. Bruce Mengi and Dr. Sarah Gravem, examines how sea star die-offs affect the balance of life in intertidal ecosystems. Today, Nathan is going to walk us through his research findings and share how he got interested in marine science and reflect on why it's so important to support students pursuing careers in the sciences, especially at this time. A bit more about Nathan. He's originally from Seattle. He loves being outdoors, and wouldn't you know it, his favorite animal is a sea star. So he's in the perfect line of work. So please join me in giving a warm virtual welcome to Nathan Pethick. Over to you, Nathan.
Thank you so much for that introduction. Let me share my screen real quick and we'll get started. So yeah, I'm Nathan Pethick. I will be talking about um, my journey into science, some of the cool results from the research that I've done at Oregon State, as well as advice to potential or aspiring scientists and anyone who's interested in getting into science. I guess I would start with a little bit about me, but you just heard it, so I think I'll skip it. I guess other than that, I'm from Seattle, came here to OSU, and did some research for 3 years. But what inspired me really to get started was back in high school during COVID. I was the class of 2021, so I graduated during the COVID pandemic, and that was a pretty turbulent time to say the least. And one of the classes that I took was AP Biology. This was completely over Zoom, and a lot of the assignments that I had were pretty optional. And one of the optional assignments I had was to watch this documentary, The Serengeti Rules. And in it, it follows scientists as they, do ecological research. And one of those scientists was Dr. Bob Payne. Dr. Bob Payne was a professor at the University of Washington, and he was the first sort of person to understand the way predators exert their influence in their communities. And his famous research study involved Pisaster ochraceus, or Pisaster, the purple and orange sea stars that you see on the coast, both in Washington, California, Oregon, just along the Pacific coast. And what he did was he removed these sea stars from stretches of shoreline and from tide pools and observed what happened over years. And what he saw was that tide pools went from this very diverse and beautiful tide pool to this, where mussels are completely dominant and absolutely took over the tide pools. And I remember watching this documentary and being like, whoa, that's such a cool finding. I didn't know that like predators could do this. This was my first introduction to the keystone species concept, which Dr. Payne was the one who coined that after this work. And so I was very blown away, but I didn't really know how to get into research, how to make this a career. I didn't even know that was an option really, because I came from a family that didn't do science. My parents weren't scientists, and it was just not on my radar. However, I decided on a whim, kind of just like getting emails in from colleges. It's COVID, just like why not send out applications to places they're not doing SATs and whatnot. So just, one of the applications I sent out was for Oregon State as a biology major. And I eventually made my way here to Oregon State. And, as I joined the honors college, and, we have to do an honors thesis. And one of my friends was like, hey, you should look into this. Because I was recounting this exact story about, oh, this would be really cool to do, but I don't really know how to do it. And they're just like, hey, you should just try to see if anyone does research in the tide pools. And lo and behold, my would-be mentor now, po or my old mentor now, Dr. Bruce Mengi, he does exactly this type of research, studying the tide pools, studying sea stars and whatnot. And when I was researching him before my first meeting with him, it turns out that he did his PhD with Dr. Bob Payne. So that was a really cool full circle moment for me and kind of inspired me to keep going when, the going gets tough. So moving on to some of my work within the Mengi lab, I've been involved in the lab for about 3 years now. I've been helping out with about four different projects on a regular basis, cleaning up data sets, collecting data, all the everything involved. But there was two that I really was super involved with. One of them was on top, that top photo. I presented some of that research at a national conference in Portland, the Western Society of Naturalists back in November of last year. And then this past June, about a month ago, I defended my honors thesis. And beyond the direct research that I've done, I've also had the opportunity to mentor other undergraduates, such as freshmen just coming in, getting started with research on their own through OSU's URSA program. And I'm really thankful for my mentors to, trust me to help teach these freshmen and aspiring researchers to get involved with science. And so that was a really cool opportunity to really hone my skills and, practice teaching people about the science that I get excited about. And so two of the biggest projects, again, that I've presented on are what we call our predation rate experiments and our annual colonization experiment. So before I get into any of the actual research that I've done, both of them kind of center around this topic called sea star wasting disease. So I wanted to provide a little bit of context for that. Sea star wasting disease was this epidemic that impacted about 20 species of sea stars all along the Pacific coast of North America, from Baja California all the way up to Alaska. Um, in Oregon specifically, Pycnopodia, they're these big 20-armed sea stars. They were impacted pretty heavily. And then Pisaster, those purple and orange ones you see in the tide pool, they were also hit pretty hard. And so those two species were the ones that were impacted the most in Oregon. And I focused mine on Pisaster because those are the ones that we find in tide pools, whereas Pycnopodia are more subtidal. And so if you look at the graph, there is years on the x-axis along the bottom, and then the adult density, or just how many individuals are packed into a given area, in this case meters squared, on the y-axis. And then each of those graphs is a given cape, with Cape Foulweather in the north, Cape Perpetua, of course, in the center, and then Cape Blanco all the way down in the south. And I wanted to, again, provide this graph to show a little context on the devastation sea star wasting disease had, because we saw between 60 and 90% loss of biomass, depending on what site we're looking at. And these graphs show that pretty well. Cape Perpetua and Cape Blanco both, I think, show that the most extreme. And so you can see prior to 2014, that red line, there is a relatively high density of Pisaster adults, sea star adults. And then once it hits that 2014 red line, it just crashes in all three capes at all 6 or 7 sites. And it just really shows the devastation and how it lasted for so long. And because it stays low until about 2020, 2021, where we finally start to see that uptick. And so that uptick is what I was really curious about. What changes happened in between the pre-wasting disease phase and the recovering the post-2020 phase. And so that's where my research centers around. So one of the first ones that I did, the talk that I gave at the Western Society of Naturalists, went around this question of how were sea star feeding habits affected by wasting disease. So to really answer this question, we used this experiment that we call predation rate. And so we set this up in the intertidal. We have the fence treatment in the picture that you can see. It just has two sides on it. And then we have our cage treatment, which acts as a control, and it has four fully fenced in sides. And so the control doesn't allow sea stars in to eat the mussels, whereas the fence allows sea stars in to eat the mussels. And we're able to compare the rate of loss of mussels because one, obviously, sea stars can eat, and the other one, sea stars can't. And so by comparing those two, we get a good idea of how many mussels are being eaten by sea stars, which is important to understand biodiversity as mussels take up space, but when they're consumed by sea stars, they leave bare space in their wake, and that bare space can be used by algae or barnacles or anemones to increase the biodiversity and resilience of the ecosystem. So really understanding how these dynamics play, and especially in the wake of a big disease epidemic like we saw with wasting, is really important. And so jumping into some of the results on the top graph, it's very similar to that one that I just showed you, where we have years on the bottom, and then on the y-axis along the up and down, we have predation rate, or just the average number of mussels eaten per day. And so it again shows a pretty similar story to what we saw with the densities of adults. Prior to 2014, they were relatively high. Cape Perpetua and Cape Blanco both show that pretty well. And then there's that crash in 2014, and they stayed quite low until, again, 2021 or whatever, 2020 or 2021, excuse me. And then we start to see, well, at least it looks like there's starting to be a recovery in the amount of mussels that they're eating per day. And so to really analyze that relationship better, there's that graph on the bottom, and that's breaking it into phases. So instead of just having years, we assigned a cluster of years to a phase. And so we have the before phase, which is 2007 to 2013, the during phase, which is just 2014, the post, which is 2015 to 2020, and then finally the recovering phase from 2021 and onward. And this graph really allows us to compare how the points in the before phase and the recovering phase compare to each other. And the only two sites that we didn't see recovery, or at least what we would deem recovered, was at Strawberry Hill in Cape Perpetua, and then Cape Blanco, the site in Cape Blanco proper. And so this again showed that four of our six sites that we were studying showed that the number of mussels being consumed per day had gone back to the pre-sea star wasting disease level, which was a pretty exciting result to see. And this project and working on this kind of led me to finally develop my thesis. And my thesis centered around this question was, how did the prey communities instead of the predators, how did they respond to wasting disease? Before I get into the results again, I want to kind of go over some of the field methods that we used. For this, I actually used a couple of different things, both in the field and in the lab. In the field, we used these belt transect surveys. Basically, we put out a 5 meter long tape, we put it below the mussel bed, and then collect every single sea star that we see 1 meter above and 1 meter below the tape. You can see a pile of them waiting to be weighed. We weigh them, and then we measure how long their arms are. Then we also have our ACE experiment, the annual colonization experiment, and it's very similar to predation rate, where we have cages and fences, but instead of looking at how the prey are being consumed by predators, we're looking at how barnacles and mussels compete for space, as well as how predators like Pisaster might be influencing that competition. And so also in the field, we take photos of the ace cages to later analyze in the lab to understand who's there, who's not, and how much space they're taking up in the cage. And so when we get to the lab, I import the photos that we take in the field to a program called PhotoQuad. It looks a little bit like this. Then I draw over a plot like that, and then I'll overlay 100 points. And then after I overlay the 100 points, I just simply choose what species are present underneath that point. And that gives us an idea of maybe there's like 20% barnacle in this photo. Maybe there's that one mussel that's taking up about 3% space. There looks like there's a limpet, stuff like that. And so that allows us to understand who's there and who isn't. Jumping into my results, but before looking at the actual prey species, we first wanted to understand how did the sea stars change? Because we know that there's the mass die-off. However, we don't know what happened after. We didn't know exactly what happened after that. And so this graph, it shows on the bottom the weight of a sea star, and then on the Y there is the number of sea stars observed, And then for each of those long rectangles, that is a given year. So starting in 2000 all the way to 2024, we have data of just a distribution of the sizes of sea stars that we saw. And so you can see from 2000 to 2014, we have that classic um, adult-dominated size class at our sites. And then when C-star wasting hit between 2014 and 2015, the adults mostly died out, and then we had a giant recruitment event of little baby sea stars, between 7 and 300 times greater recruitment than the year prior. So we had an unprecedented die-off of adults, followed by an unprecedented recruitment of babies, which is really interesting, and we actually don't know what caused the recruitment event, and we're actually still trying to figure that out. But it really changes the picture because a baby sea star, like a infant, isn't going to eat a whole lot compared to an adult sea star or like a 21-year-old college student, There's going to be a big difference in who's consuming what and how much they're consuming. And so that really gets at the question of, how did the prey communities respond to this change in just the demographics? For my thesis, we did some fancy statistical modeling and we wanted to understand who was most abundant, and we gave it four different population structures: a large adult dominated, an adult dominated, a juvenile and recruit dominated population structure, and that basically is just telling us who on average was most abundant at that site at that year. And so you can see that it goes from large adult and adult dominated to that crash of recruit dominated. But I think it's pretty cool that you can see it go slowly from recruit to juvenile dominated. Now we're starting to see the sites become adult dominated, showing that recovery. And so now with that in mind, we can jump to actually understanding how the prey responded to that change in demographics to the recruit and juveniles, and if there were any roles in the intertidal with the juveniles and recruits. And so our first graph here on the left, that is looking at how the prey changed over time. So what we predicted with an increase in juvenile and recruits, they would go up higher. We know they like to be higher up above the muscle bed compared to adults who like to be below the muscle bed. And so our ACE experiment is up in the upper end of the muscle bed. And so we assumed that within the huge influx of recruits and juveniles, we would see a decline in prey over time. So that's what we have: time on the Y, and then percent cover of prey or year on the X, and then percent cover on the Y. And so we would expect the negative trends that we see at Fogarty, Boiler Bay, Yachats for mussels, and then at Rocky Point. That negative trend is what we expected to see. We expected as there's more juveniles and recruits, they would consume those smaller prey items, and over time, those prey would decline on average. However, to answer was it the recruits that were actually causing this, the graph on the right helps us answer that. It's the same setup as with percent cover on the Y. However, we're changing the X to have mean biomass density. So it's similar to that very first graph that I showed you, the number of sea stars or the weight of sea stars crammed into a single area. And we go from 0 to 1,000 grams per meter squared, where 1,000 grams per meter squared would be, imagine 1,000 grams of sea star in 1 square meter. You would imagine that they would be pretty hungry, that many sea stars, 1,000 grams of sea stars compared to about 10 grams per meter squared. And so we expected to see the same sort of negative trend that we saw in the left graph. However, Fogarty Creek, Boiler Bay, Yachats, Strawberry Hill, and Cape Blanco all show a positive trend, meaning that as there were more sea stars present, there was also more prey, which is very counterintuitive and was not what we expected, because that to us means that sea stars were helping prey be there, when in theory, prey should be being consumed by sea stars. And so that was a bit of an unexpected result. In my thesis, I touched on a couple of different alternative explanations for that, but we haven't really started to dig into that yet, and it's an area of future research. And so just a good take-home point is that we observed declines in prey abundance, but we couldn't attribute that decline to sea stars directly. And so, with all this, I had been very involved with the lab and at the time finished my thesis last month. And, it's been since then, it's been a good time to reflect on my time that I've had in research as an undergrad, getting started in the end of my freshman year all the way through graduation. And so I think the very first time I reached out to Dr. Mengi, I was very nervous. He's a professor, he does all this cool research, and I didn't even realize that there were different species of mussels and barnacles, and I couldn't even tell you what the scientific name for a sea star was. I didn't think that we even had anemones in the Pacific Northwest. So to me, I felt like there's no way he should be meeting with me. I shouldn't be doing this. And you know, it surprised me when I got through my first meeting how encouraging he was and how eager he was to take me on as a student. And I think that was, at least as someone who didn't have any sort of time spent in science, and no anyone to follow in science. I think that was a big shock. And that's, I think, one of the biggest reflections that I can take, besides just how welcoming and eager people across all fields of science are for, undergrads and for people to explore their passions and to find what could be their potential career. And so I think the big reflection is just how how kind everyone is in science, at least in my experience. I know there's going to be some bad apples, just like everywhere in life, you're going to have some bad apples. But it was a real shock to me. And from someone that was, a scared freshman, being so supported in my lab all the way through from freshman year to graduation, I think that was a really good thing. And that in turn makes me want to give advice for aspiring scientists. Maybe someone here is just going into college, you have your siblings, your child, your grandchild, your friend, whoever that wants to get started in science and might have been similar position as me and doesn't know the sort of steps to take to get in there. And I would say just, get out there and email a professor if you're starting college. If you're already out of college and you want to like get involved in science, reach out to local organizations and just ask if you can volunteer and help out with them. I think that's the biggest thing, and I think that was the biggest thing to get me started was my friend, being like, you should just email someone and see if they do that sort of research. And so if it wasn't for that, I don't think I would have been as confident and as excited about science as I've been. And I don't think I would have gotten the opportunities that I've gotten if someone didn't give me that little push. And so that's my big advice, I guess, is to, it might seem like a big leap, but in the big picture, it really isn't. And just getting out there and just putting your feet in the sand, I think that's really important to get out there and to develop your passion in science. But also, I would be remiss to not mention the state of science today. This article from the New York Times analyzed the funding of National Science Foundation grants over the last about 10 years compared to this year, along the same period. So the first 6 months of funding for those years compared to the first 6 months of funding this year. And we've seen we've seen about $1 billion decrease in funding. And it's really sad to see the lack of funding now and today in science, given how many advances that we have through science, like pretty much everything from medicine to physics to engineering to technology. You see it right there in front of you. The amount of money that went to all these fields and how all of these fields have brought us to where we are today and advanced us to where we are today. And to go back and to lose that sort of funding, go back decades in the amount of funding that we're giving these very important ventures, I think it's important to both advocate for science, but also advocate for people to get into science because scientists are going to advocate for science more than anyone else. I mean, I think that's just kind of a fact of life. We're going to advocate for our field, but I think encouraging students to get into science as well, that might seem counterintuitive, but getting them out there and because we need more scientists. And it shows the people in power that, hey, this is an important thing for us. We care about this. We want science to be funded so we can advance technology, advance medicine, advance climate research. And so I think it's really important to not just get into science, but to advocate for science, especially today. So, with that being said, thank you all for being here and for listening to me today. Um, Thank you so much. Are there any questions?
Great. Thank you so much, Nathan. I really appreciate the note that you ended on. I wholeheartedly agree that we need to support those in the sciences currently and those looking to have careers in the sciences and not get too discouraged by what they hear and see in the news. We are always going to need people like you doing this kind of work and monitoring our environment and finding out weird things like when your experiment or the results didn't go the way you thought they were going to go. That's a interesting question to try to figure out, in the next iteration of this project. So I'm gonna go through a couple questions that we got uh, in the next few minutes that we have. Uh, William Hans Schumacher, I apologize if I mispronounced that. He asked, what is the current theory for the root cause of sea star wasting disease? If
Yeah. Oh, sorry.
Go ahead.
So the common knowledge right now is that it was a virus. However, I'm aware that there's a paper that's under embargo right now. So it's coming out soon, but the authors can't talk about it yet until it's published.
Oh.
And so they offer an alternative explanation beyond a virus. I don't know what their explanation is beyond that it's not a virus, but until now, we've been told that it was a virus. So I'll say a virus. I haven't read the paper. I don't know if it's out yet, but I've been taught for the past 3 or 4 years that it was a virus. But maybe if you get back to me in 3 to 6 months, I'll have a different
Yeah.
answer for you.
I'm so curious. Like I'm like, what other explanation could there be
I know,
besides
I am too.
virus?
There's always science coming out, so we'll
Right.
see.
Because for those, I know a lot of us are familiar with the sea star wasting disease, but do you want to describe what it does to a sea star or what we think, if it is a disease, like if what we observed in sea stars?
Yeah, so you would have like your normal healthy sea star, um, and they look, healthy, but then they get sea star wasting disease. They go from healthy, they grow lesions, little like white spots, kind of like I don't know, a blister or like a scrape that you would get, and they would scab over. They would get these white lesions, and then slowly their arms would start to literally just disintegrate. And they would just die like that because unfortunately they're, they're different than us. And so when they start to disintegrate like that, there's no real coming back from that.
Yeah, it was I think you mentioned earlier in the talk, it was like 50 to 90%
Yeah.
fatality. Like that's a crazy kind of pandemic that the sea stars had. So, yeah, it'll be interesting to see if there's another explanation for it. Um, oh, Paige Faust, one of our other young wave makers, she asked the question, what are your next steps in your
I
career?
will be going down to California at Cal Poly San Luis Obispo to join my co-mentor Sarah Gravem. Down there, she'll be starting up a lab. She'll be a faculty there, and I'll help her, start up her lab for the first 6 months or so for a faculty research assistant position for 6 months. So very excited for that.
Yeah, that's incredible. I'm so stoked for you that you got that opportunity. I think you're gonna have an amazing time and it'll be an incredible experience for you in this field. So yeah, congrats on getting that.
Thank you.
We have one last question from Maureen Focal. She asks, how can our team of teachers volunteering for Coast Watch collect data in a tide pool? Is water quality something to consider? So forgive me if I misconstrue this, Maureen, but I think what she's asking is like what sort of things should you consider if you're collecting data in a tide pool? Like what sort of variables should you be looking at?
Yeah, so tide pools are going to be different from like the actual waves. And so at least for us, we use scientific instruments to get measures of oxygen content, the level of chlorophyll or nutrients in the water. But if you're just out there tide pooling, you can use iNaturalist to take photos of what you see. If you run into Pycnopodia, those big 20-armed sea stars, if you find one in a tide pool, definitely take a picture of that and post that to iNaturalist. Sarah Gravem, she also is looking at community collected data to understand where Pycnopodia are, especially in Oregon and California. And so taking pictures of what you see, uploading it to iNaturalist is one really good way to help out and engage scientists. But more on our side of stuff and water quality, we more use scientific instruments to do that just because of how technical that is.
Yeah. Yep. All the nitty-gritty sort of variables require very specific instrumentation. But yeah, just getting some ideas on the number of animals in a particular tide pool. Can definitely use iNaturalist. I'll just put a plug for an event that Cape Perpetua Collaborative is hosting soon on this Saturday, July 19th at the Cape Perpetua Visitor Center. We're actually leading a class on how to use iNaturalist. So that'll be at the Cape Perpetua Visitor Center this Saturday, starting at 1 o'clock in the afternoon. And I believe we are going to have to shoot off here pretty soon. I will pass on other questions to Nathan um, and get you guys in contact. So I apologize that we've run out of time, but it just sort of speaks to how interesting this topic is and how many people are interested in what's going on with our sea stars and our tide pools on the coast. So thank you, Nathan, for sharing your work with us today. And thank you to all who joined us today. If you want to share Nathan's talk, I will be posting it to YouTube. I also still need to post Alexis's from last week, so there'll be a couple of YouTube episodes coming your way. And these will also be available in a podcast format very soon. So thank you again, Nathan. Thank you to everyone who attended today. And we will see you next time at our next Young Wavemakers talk.
Thank you
Bye,
so much for having
everybody.
me, and thank you so much for coming, everyone.
Bye.